Imagine you can store every picture you've ever taken, every song you've ever listened to, and all 14 seasons of Grey's Anatomy on a DNA size so small that you can't even see it. Well, this is the first step in that direction. So first I'm going to explain the limitations of current data storage methods. We are estimated to generate 44 trillion gigabytes of data by 2020. You can think of a gigabyte like each phone has something like 16 to 64 gigabytes. However, DNA storage capacity is far beyond digital storage capacity. It's around 1 zettabyte, or over 34 billion smartphones worth of data. Some other reasons why DNA makes for good data storage is that it can remain stable for 100,000 years. It also won't grow obsolete like a CD or a hard drive. It doesn't require a power supply to be maintained, so it's less likely to have technical failures. You can't exactly wipe out DNA the same way you would a hard drive. It's also small, easy to store and transport. So you've already been seeing that we've reached the limitations of size for hard drives. So this is a good alternative. So my paper is called The CRISPR-Cas encoding of a digital movie into the genomes of a population of living bacteria. It took place at Harvard and it was published in Nature. In other words, storing a GIF in bacterial DNA. So the main idea of this paper was trying to see if you could store data in living bacteria. They have already stored data in synthetic DNA. For example, they stored all of Shakespeare's sonnets in DNA. However, this is the first time they've done it on living bacteria. And living bacteria has additional problems because they're moving, changing, dividing, and dying. And also, this was the first time they had stored a picture and a video. So what they did is that they encoded the pixel values of a black and white image, and then later a short movie, into the genomes of E. coli bacteria. So does anyone have any questions on this concept? Okay. So the overview of the process. The first step was actually converting the pixel values into the nucleotide codes. Then the second step was actually making the DNA, these oligonucleotides, which just means short DNA fragments, for each of the nucleotide codes. And then the third step was electroporating, which I'm going to explain later, these oligonucleotides into the bacteria that have these CRISPR arrays. And they stored it in 600,000 cells to increase accuracy, but you can imagine that these cells are very small, so that even though that sounds like a big number, it's not actually that big. And then at this step, the DNA gets acquired into their genomes, and then lastly, they can recover the information and restore the picture by sequencing. So I'm going to walk you through each step now. I think it actually missed a slide. Oops, sorry. This is So these scientists were able to utilize bacteria's natural immune system, called CRISPR-Cas microbial immune system. And bacteria naturally use this system to store the nucleotides of invading viruses to build up immunity. And this way it allows them to recognize and attack these viruses in the future. However, in this case, the scientists were able to utilize it in order to make their own DNA that looks like the viral DNA. And it would get incorporated into the bacteria's genome the same way that viruses would. And they did this in the form of oligonucleotides, which just means short DNA again. And you can think of it as an array. And each of the DNA sequences have to be 33 base pairs or nucleotides long. And there's two main proteins involved, Cas1 and Cas2. So does anyone have any questions on the CRISPR-Cas9 system? So this was the original picture they decided to store in DNA. It's 30 by 30 pixels and only 494 bytes. So for comparison, a gigabyte is a billion bytes. So this is a very small image they're storing. And you can already make out the different pixels that they're going to store in sequences. So the first step was actually converting the pixel values into nucleotide codes. So what they did is they split up this picture into 21 pixel colors, like varying shades of white and gray, and they made them into nucleotide triplets. So you can see here on the right is a chart of how they coded this. So the first pixel would end up being TTT, like to the left, to the top, and to the right. Second one would be TCT, and so on. They only use 21 colors, but you see that this chart gives room for much more than 21 colors, and additionally you can end up using more and more nucleotides, and you could code as many colors as you'd like. So the second step was actually making the DNA, these synthetic oligonucleotides. So I'm going to walk you through an example on the left, the sequence. So the first two letters were the AA, and that was what is called a protospacer adjacent motif, or PAM. It's not that important, but basically in past studies, scientists have found that this sequence will help increase acquisition into the genome. Then the next four are what's called a pick set. So as you can see, they divided up the picture into 9 boxes. And the pick set helps specify which box the sequence belongs to, so that at the end, when you're reconstructing the image, you can put it back into those locations and recreate the picture in that format. And then after that, there's 28 bases remaining, and since each pixel was coded into 3 nucleotides, this can approximately store 9 pixels. And on the left, you can see a small example of how they converted the binary pixels into the nucleotides. And on the right, that hairpin formation is just the formation of the DNA they stored them in. Does anyone have any questions on this?
Could you just walk through the whole process again?
Yeah, of course.
Sorry,
It's pretty
I
complicated.
did not get it.
So the first 2 letters are what's called a protospacer adjacent motif. It's not that important, but basically it just helps increase acquisition into the genome. Then the next 4 are what's called a pick set, and it basically just specifies where in the picture those sequences encode for. So when you're reconstructing the image, you can put them back in those locations. And then lastly, there remains 28 bases left. And since they converted each of the pixels into 3 nucleotides, that approximately can store 9 pixels. And then they have many of these sequences. This was just one.
Anjali?
Yeah. Oops,
Do you know why 28? Was it just that was the optimal length of DNA, or was there some other reason?
So the CRISPR array splits them up into these spacers, that's what they're called, and those spacers are 33 base pairs.
Okay.
So they had to work around that, and so these were 33.
Thank you.
Step 3 is electroporation. What this is using electricity in order to open up the pores in the E. coli membrane, so the DNA can flow through and interact with the Cas1 and Cas2 proteins of the CRISPR array. So in the first step, you can see they used electricity. Then in the second step, it's interacting with the Cas1 and Cas2 proteins and incorporate it into the CRISPR array. And then they use sequencing to recall the image, and you can see the image on both sides because they reconstructed it. So step 4 was recovery. The bacteria continued to replicate after being incorporated into the genome, and the next day they sequenced it. So they used a computer program to decode all the nucleotides back into pixels. So you can see here that after receiving all the nucleotides, they're able to go back into the pixel versions. And then they used the pick sets in order to rearrange the picture, like where they were located. So this was the original image, and this was the reconstructed image. So they're pretty similar. Sounds easy enough, right? Except that this took 655,000 reads. A read is just a simple single time sequencing it of bacteria. So this was clearly a long and inefficient process. However, they were able to recover 96% of pick set sequences. So at 10,000, you can see that there were still many black spots that hadn't been accurately recalled yet. At 20,000, there were fewer black spots, and at 40,000, it's near perfection. But after many more thousand reads, they were able to gain 96% accuracy. And in terms of time, this took approximately 2 hours and 40 minutes to gain near perfection. But at 10 minutes, you can see that they started being, the spacer acquisition could be seen at 10 minutes, and then over time, more and more pixels were able to be recalled. So as you can see, some spacers were acquired faster and more often than other spacers. So they wanted to understand why that was. So they looked at the sequences of all these spacers that were acquired faster and found some commonality in the ends of them. So on the top, you can see the spacers that were the sequences that were most likely to be acquired. At the bottom, you can see the ones that were least likely to be acquired. So the G's and the A's, for example, are more likely to be acquired. And they called this the acquisition affecting motif. So the ends of the nucleotides clearly have a part in how fast it's being acquired into the, yeah.
So the x-axis piece here, that's the length of the DNA. So like you're looking at the base in that spot? Or what's the spread here?
Yes, I believe that's the size of the DNA, and then the ones that are on the top are the overrepresented sequences, and the ones on the bottom, the underrepresented. So you can see the G and the A at the end they really like that, and that gets acquired really fast. So they're able to use this in the future when they're actually incorporating like important data, because then they could put it in this format and be more likely to be acquired. Any other questions? Okay. So now they wanted to see if they could store a movie or this short little GIF in DNA. So they ended up picking this short movie of a racehorse. And a little backstory to this video is that it was the first ever motion picture, and now it's going to be the first picture ever stored in movie in DNA. So it has that fun fact. And it was taken by Edward Muybridge in 1878 because he wanted to see whether horses ever had all 4 feet on the ground while running. So you can come up to your own conclusion about that. And it's 36 by 26 pixels, so it's still not that high data, totaling 2.6 kilobytes. So it's a lot more than before, but again, they still haven't tested on much bigger amounts of data. And a gigabyte is approximately 1 million bytes, or 1 million kilobytes. This is more than before, but still very little. So the first step was, again, converting into nucleotides. So instead of 1 picture this time, it was 5 separate frames of this galloping mare. And this time, instead of putting the pixel at the front, they put it at the end. And they have that GA at the end, as we learned, helps increase acquisition. And this time they, again, split it up into 9 boxes and had pixels saying where that was located. However, they didn't have nucleotides specifying what frame it was. So it seemed like no way to know whether it'd be the first frame or the last frame of the mare. But, does anyone have any questions really quick? Okay. But they did find a way to determine the order of the frames. So what they did is they electroporated each frame over 5 days, with about 18 hours in between them. And these were recorded in reverse chronology of how they occurred. So whatever spacer they electroporated first would end up being the furthest away at the end. So this is important for future because it provides timestamps of when the data was acquired. So when we're starting to use it with like real uses, we'll know that this was acquired before and this was acquired later. So they were able to recreate the order of the frames. And this was the original versus the recreated. So as you can see, there's many of these like black spots, but for the most part, it's pretty accurate. So this time it took over 4 million reads. It's a lot more than 600,000 before. And they were only able to gain 90% accuracy. So it takes a lot more time, it's less accurate, but this could eventually become a mainstream use of data storage. So you can see at 5,000, there's many black spots, and by 4 million, there's a lot fewer. And that was the result. So there are some downsides to using DNA. For one thing, it's very expensive. It's around $12,400 per megabyte of data. So that's very expensive, and it's not going to become mainstream anytime soon, but eventually could be with more research. And you can see that's also a very slow process. It took some thousands of reads to regain the picture. Also, DNA isn't easily rewritable the same way you would a Google Doc. If you want to update part of the information, you have to redo all of the information storage process. It also doesn't allow for random access, meaning if you want to access part of the data, you're going to have to decode all of it. And lastly, with DNA comes risk of mutations. So while this didn't play a part that much in this case, over many thousands of years, it's probably going to play a part. However, the bigger picture is that this is a whole new direction of data storage, and it could eventually become a common form of data storage. DNA is special because it's small and has a large data storage capacity, and the Harvard scientists predict that you might actually be able to store data in your skin someday. So like imagine storing like your favorite movies in your skin, that'd be kind of crazy. And it has many applications even in the science world. For example, they could program microbes to monitor environmental pollution. This is important because it provides timestamps of when the environment started changing. And also, they could program neurons to record how the brain develops and communicates in a living animal. So we could learn how neurons develop and how they communicate with other neurons. And we currently don't know that much about the brain, so this is like a whole new idea of how we could get some inside scoop on what's going on. And another option is like, for example, when someone gets sick, what if there's a way that you could like go back and see what got them sick in their DNA. So that's one option too. And this study also helped us better understand the CRISPR-Cas system and its data storing capacity. In particular, they found sequences that increase acquisition, and they found a way to determine the order of acquisition. So that's important for the future because we can use that next time we try new research. So does anyone have any questions? Yeah.
Can this only be in black and white, or
Well, this one was in black and white because it was a very they were trying it on a very small scale, but it could eventually become more colors, I imagine, because it's not actually the colors that are being stored; it's just the sequence of the pixels. So if you had other colors, you could just have more nucleotides specifying those colors. Yeah. So, thank you.